Have you ever wondered how your metabolism works after food intake? A balanced diet is based on the intake of suitable amounts of different types of nutrients, such as sugars, lipids, and proteins. The sugars we ingest in our diet present different levels of complexity. Monosaccharides, such as glucose from fructose, disaccharides, such as lactose from milk, and even complex polysaccharides, such as starch and glycogen. During digestion in the gastrointestinal tract, all of these sugars are converted into the corresponding monosaccharides. These are absorbed through the wall of the small intestine and enter the bloodstream through the portal vein, which transports them to the liver for distribution. Among the monosaccharides, glucose plays a fundamental role since it is the main source of energy for the brain and the only source for red blood cells. The nervous system, testes, renal medulla and embryonic tissues also use glucose as their main source of energy. In fact, the maintenance of blood glucose levels within a certain range is essential for our health. The liver is the organ in charge of transforming and distributing most of the nutrients ingested by adjusting its metabolism to the different nutritional situations to ensure the availability of nutrients and precursors to the rest of the tissues and organs, including glucose for brain function. In fact, the maintenance of blood glucose levels depends on the metabolic flexibility of the liver. The metabolism of the different tissues must be coordinated to meet the needs of the organism. This control lies in the neuroendocrine system and is mediated by the action of hormones that regulate the key enzymes of the various metabolic pathways. The hormones, adrenaline and cortisol, produced by the adrenal glands, adjust metabolism in stressful situations. On the other hand, glucagon and insulin, produced by the alpha and beta cells of the islets of Langerhans in the pancreas, adjust metabolism to situations of low and high levels of blood glucose, respectively. Subsequent to food intake, glucose levels increase in the blood. This triggers the entry of glucose via the glucose transporter 2 into the beta cells of the pancreas. ATP generation from glucose metabolism in these cells triggers a signaling cascade that culminates in insulin secretion. This molecule travels through the blood into cells where it binds to receptors on different cell types and triggers signaling cascades to control metabolism. The entry of glucose into the hepatocyte is favoured by the release of hexokinase 4 from its sequestration in the nucleus. This enzyme phosphorylates glucose, which allows unlimited entry of sugar into the cell. The liver then acts as a distributor of glucose to the rest of the body to meet the energy needs of the brain, red blood cells, and muscles. However, once certain blood levels are reached, the release of glucose from the liver stops. What happens to the glucose if it exceeds our body's needs? The liver uses this excess to replenish its glycogen stores. The function of liver glycogen is to maintain blood glucose levels between meals. This polysaccharide accumulates in limited quantity in the liver, and hence its levels at a given time depend on the time elapsed between the previous and the current meal. Glycogen synthase, an enzyme that regulates glycogen synthesis, is activated by insulin-triggered dephosphorylation. Furthermore, insulin inactivates GSK3, which is responsible for the phosphorylation and inactivation of this enzyme. In the same way, the muscle also synthesizes glycogen with the excess of glucose in the blood. This glycogen, unlike that stored in the liver, is used by the muscle to obtain energy quickly when needed. Once hepatic glycogen stores have been replenished, the liver metabolizes excess glucose first into pyruvate by glycolysis and then into acetyl-CoA by the action of the PDH complex. Both processes are most active in the presence of high blood glucose concentration. The binding of insulin to its receptors activates protein phosphatase 1, which promotes the activation of PFK1, the main regulatory enzyme of glycolysis, through the increase of fructose 2,6-biphosphate levels. Therefore, the concentration of pyruvate increases, which maintains the PDH complex active, thereby preventing its phosphorylation and consequent inactivation. What is the fate of this acetyl-CoA? When glucose levels reaching the liver are very high, acetyl-CoA is used for the synthesis of fatty acids. Insulin-regulated acetyl-CoA carboxylase is essential to maintain this enzyme in its active dephosphorylated form, capable of polymerizing and catalyzing the initial reaction of fatty acid synthesis. The lipids synthesized by the liver are packaged as lipoproteins and transported to the adipose tissue for storage as triacylglycerols. In the same way, these lipids also reach the muscle, where they are completely oxidized to obtain the ATP necessary for muscle contraction. These are the reasons why the functioning of the liver after feeding is called lipogenic liver. Obviously, the amount of fat that is synthesized depends on the levels of carbohydrates in our diet. Therefore, a balanced diet is essential for the well-being of our organism.